Well, it's a very great pleasure and I think rather an honour for me to be standing here in front of you today, and I thank very much our organisers for the invitation that I received to come here and talk to you about the work that we've been doing recently over the last 10 years, I would say, in Nantes in France, on the mechanisms, the natural mechanisms by which deuterium depletion occurs in natural products, in metabolites. And I really want to thank particularly my colleague Isabelle Bilot, who did a great deal of the work that I'm going to be presenting to you today, and other colleagues, one of whom, in fact, was Benlizon, who we were talking about earlier. I notice we're actually drinking deuterium-depleted water. So all the numbers I give you, obviously from now on, have to be a few ppm lower. Okay, so I just want to do a little bit of background, and just to remind you again that metabolites, when they're just looked at in nature, have different deuterium levels. We've just heard very clearly and very nice presentation telling us about how water deuterium varies, and I'm going to talk to you about how biologically incorporated deuterium varies. And just to put a little bit of numbers on this, here's a nice 2D plot of, we have this delta deuterium on the axis here, and I've put exactly the same thing but for carbon-13 on the other axis, just to spread things out a little bit. But we're really interested in variation on the deuterium axis here. And let's just look at a few metabolites. Here's cellulose, the most common organic compound in existence. And we see that cellulose has considerable variation depending on the source that it comes from. The C13 variation is due to the metabolic nature of the plant that is producing the cellulose, but the deuterium is related very much more, for example, to this polar equatorial difference that we've just been hearing about in the source water. If we look at one or two other compounds, let's look at sucrose, the primary metabolite, and starch. And we see here again the typical carbon-13 separation, but we notice also here that there is in fact a deuterium effect depending on which type of plant metabolism is involved in the synthesis. Now, if we take that sucrose and we ferment it to ethanol, we see straight away that there is a significant change in the deuterium content of the metabolite that we're looking at. In this case, we've got a move of about 150 ppm delta in deuterium. And the same very much if we take sucrose with C3 and we ferment it to ethanol. So this difference here is maintained in this instance in the metabolites produced by the fermentation of our sugar. If we take another product, a secondary metabolite, vanillin, a phenolic compound, which many people will know, anybody that eats ice cream will know about vanillin, was also much favoured of perfume at one point. Vanillin has different sources. Here's vanillin from the plant that makes vanillin, Vanilla planifolia. This is a C3 vanillin from another sort of plant. And if we look at this fossil vanillin here, this is actually produced from petrochemical sources, we see again a huge spread on the delta deuterium scale. So why do we get this in metabolism? Okay. Well, if we just look at a plant, and we've heard a little bit about this already, we can see here that the leaf water, as we've already heard in plants, is relatively high. When we start looking through the metabolic cycle of plants, and don't worry about the detail on here at the moment, I just want to make the principle. We have two main ways, origins of the hydrogen that becomes incorporated into compounds in plants. Colours changed a little bit here. These were red, but they've become brown. Perhaps that's better for plants. We have two main sources. We have a very key metabolite, pyruvate, and as we see, these compounds are lower in deuterium than the source leaf water. Pyruvate, of course, moves through various metabolic pathways into compounds, such as the soluble carbohydrates. And here we see these are richer than pyruvate. And that's perhaps not surprising, because pyruvate, of course, is a metabolite produced from the carbohydrates. So here's that same fractionation as I showed you for ethanol. If we look at the fatty acids, again, produced from pyruvate, here we have a further depletion in deuterium during the biosynthesis of the fatty acids. And this is characteristic of many metabolic pathways. Essentially, the longer the pathway, the more depletion in deuterium you get in the end products. Anyway, I put blue rings around these two groups of metabolites because these are what I want to use to illustrate to you biological fractionation and means within metabolism that natural deuterium depletion is produced. Now I can skip over this one very fast because we just had a nice talk about it. And the physical effects of deuterium depletion I can move over straight away, and I can move on to the metabolic effects that introduce deuterium depletion. And I just want to go through a couple of concepts with you. Some of these, I'm afraid, will bore you to tears for the people that are familiar with it, but just in case there are a few people who are not so familiar with isotope fractionation and kinetic isotope effects, I'd just like to fill in the background because they're fairly important for some of the things I'm going to talk about in the next half an hour. I'll try and keep it half an hour. Basically, if you have a deuterium in a position in your substrate which is important for the reaction to take place, in most cases, and I say most cases, it's not always true, in most cases, breaking the deuterium carbon bond or the deuterium nitrogen bond requires rather more energy than if you're breaking the deuterium hydrogen bond. Now, I've drawn this graph slightly fictitiously. In fact, the transition state energy is the same, and strictly, in fact, it's the initial energy, of course, which is differs. But it's more illustrative if we look at this. So the carbon-deuterium bond needs a little bit more energy to break it, and therefore the transition state is a little bit higher, and therefore, of course, the probability of the kinetic rate with the deuterium is therefore a bit lower than with the hydrogen, the protium, and therefore we select against deuterium in this particular reaction. To put an example on that, this is an example, in fact, of a reaction in fatty acid biosynthesis that we're going to come back to later. We have two types of isotope fractionation. The first one is the primary isotope effect. And this is where we are actually cutting the carbon-hydrogen bond. As you can see in the product here, we've lost this hydrogen completely from the molecule. We've cut the carbon-hydrogen bond, and therefore we have what's called a primary isotope effect, which wouldn't surprise you, tends to be rather larger than the secondary isotope effect that we have in the hydrogen, which is retained. But the key point is that you could also have a hydrogen isotope effect on this hydrogen, which is not lost, as well as on this hydrogen that is lost. And in both cases, in the example I've shown you, this would lead to depletion in this hydrogen, which might be a small depletion, and depletion in this hydrogen, which would probably be a large depletion in the product that hydrogen is going to. In this case, we'll be talking about NADPH. The other concept I just want you to bear in mind is the concept of isotopologues and isotopomers. We're not actually interested in isotopologues today. We're only interested in isotopomers. I'm afraid I think we've lost red in the transmission. And that's a pity because a lot of my things are illustrated in red, but we'll pick them up anyway. We should have this deuterium here illustrated in red. And when I pick that up, we can see that, in fact, of course, because we have this is an achiral center until we put a deuterium there, when it then becomes a prochiral center. So we can, in fact, have two isotopomers, beg your pardon, of ethanol, where an pro-R and pro-S ethanol, depending on the position of this deuterium. Now, this doesn't become important until you become a biochemist like me, and when enzymes start working on these compounds, of course, they prefer one side to the other, frequently to a substrate, and therefore, as far as an enzyme is concerned, these two positions are not the same in this molecule, and I'll show you that in a minute. The net effect of this is basically that the physical that we've already seen, the kinetic and indeed thermodynamic effects on deuterium lead to a non-statistical distribution. In here we take ethanol. So if we have a statistical distribution in deuterium, we would expect 3 to 2 to 1, and just in the ratio of the amounts of hydrogen in each of these three groups. However, what we actually find is that the methyl group is depleted in deuterium relative to its statistical average, and and relative to the methylene group. Furthermore, this depends very much on the source of the molecule. So if we take, for example, ethanol from three compounds, and this is a deuterium Nmr spectrum, quantitative Nmr spectrum of deuterium. I'll come back and show you another example of that in a minute. Here we have the methylene group, the CH2OH. Here we have the methyl group, and this is just a standard reference compound in the middle here, which is tetramethyllurea, which we use to calibrate the NMR. And you can see, just looking at this, that the relative deuterium content of the methylene to methyl groups differ in these three ethanol samples. So we're coming back to a bit like the water we've just looked at. You have variation depending on the origin, and here we have variation depending on the origin. And here's our grapes. This is beetroot, major source of sugar, of course, and sugarcane, the other major source of sugar. And as indeed we can tell that water has been added into fruit juice, we can also tell if sugar has been added into wine, and this whole methodology was actually developed originally in our lab to stop or to show that people had added sugar into wine, which is highly illegal practice, at least in France. Okay, so the three parameters that we really need to know about, this is to measure the isotope fractionation. Now, what we're going to measure then is the isotope, the deuterium ratio, the deuterium content of each specific position, which I will be calling I or J, depending on whether it's in the substrate or the product. Here's our reference compound in the spectrum I've just shown you. That's tetramethyllurea. It doesn't have to be, and it'll be pyridine in some of the samples I show you. That's calibrated by mass spectrometry. And then we need to know the mass of the compound we've got present, both the analyte and the reference. We need to know the areas of the peaks. This, of course, is just done gravimetrically. This is obtained from the NMR spectrum by curve fitting. And we have to know the purity of the sample, and that's obtained by standard chromatographic techniques. From those, we can calculate, using a simple formula, the ratio at each position that's resolved in the spectrum that we're looking at. Furthermore, we can look at that prochirality in the case of ethanol, and there are two ways of looking at prochirality. And this is the first one, this is the simple, and this is the old one. And I'll show you the new one later on when I get onto fatty acids. Essentially, by making the mandolate of ethanol, the ethyl mandolate, here's the ethanol, here's the mandelic acid, we introduce a chiral center in the mandelic acid. And by introducing the chiral center, we can actually get a little bit of separation on these two hydrogen atoms in the methylene group. And we can separate those sufficiently well to measure the surface area under the two of them. And in this case, as you can see, the two hydrogen positions have more or less comparable hydrogen in them. But as you'll see in a minute, that's not always the case. Okay, so let's get on to looking at ethanol in a little bit more detail. It's always nice to look at ethanol in detail, especially when it's mixed up with all the other constituents that make that wine, like the excellent wine I drank last night, Hungarian wine. I'm going to look at ethanol produced by yeast. That's the standard fermentation that produces wine. And I'm also going to look at ethanol produced by lactic acid bacteria. And the reasons are, quite simply, that these two systems, these two organisms, are using different metabolic pathways to produce the ethanol. And in fact, what we want to do is to see whether we can distinguish between that by a highly complicated way of showing it, I might say, by looking at the isotopic fractionation related to the pathway. So if we take our glucose, and we've lost the red again on the sixes, but that's not a problem. It's okay, it's no problem. I'm not going to talk about the sixes anyway. If we take our ethanol, and it's fermented by glycolysis, by Saccharomyces cerevisiae, we produce two molecules of ethanol, which contain the 1 and the 2 carbons, the 5 and the 6 carbons, the 1 and the 2 and the 5 and the 6, and we lose the 3 and 4 as carbon dioxide. If we ferment exactly the same glucose by the leuconostic pathway, we lose the 1 as carbon dioxide, the 2 and the 3 come through into ethanol, and the 4, 5, 6, in fact, of course, form lactic acid, D-lactic acid in the case of leuconostoc. So if we look at Saccharomyces cerevisiae, and there's a lot of data on here, I don't want you to try and take it all in, I just want to bring out 2 points to you. What we do is we look at the fermentation in the presence of 2 slightly enriched things. Sorry, we're not looking at deuterium depletion here, we're going to enrich it very slightly and then deplete it again. This is the fermentation water, and if we take, I'll just take one example for you, and if we look here at the water, we've upped the water, here's Nantes water, nicely at 150 ppm, just about exactly where it should be, according to the talk we just listened to, west coast of France. If we double that approximately, what we can see is that the methyl group responds. The methyl group is now slightly more enriched in water, in deuterium, than it was in the control fermentation. We see that, however, the methylene group has had a much bigger effect on the deuterium content of the methylene group. And if we now look at those 2 positions separately, as I've just shown you, we see that there's a bigger effect on one group than on the other. In other words, these 2 positions have not responded exactly the same way to deuterium. And the other thing we notice, of course, is that these positions are all depleted relative to the water that was in the system. If we now do exactly the same thing with the substrate, with the glucose, we can take the glucose and we can very slightly enrich that in all the positions. There's no point in doing the 3 and 5, those just end up in water anyway. But let's just look at the 4, which is the one that is of particular interest because that passes through the reduced nucleotide pool. If we enrich the 1, of course, we see enrichment in the 1 in the methylene group, which is here. The methylene group, this becomes the methyl group, not surprising. No enrichment in the methylene group, not surprising, because that hydrogen can't be found in the methylene group of the ethanol. The 2 also enriches the methyl group, but not the methylene group. Compare the control here with the value. I'll come back to how that does it in a minute. So this hydrogen, in fact, enriches that group due to an intramolecular transfer. The 4 position is perhaps the most interesting. If we enrich the 4 position very slightly, what we find again is, as we'd expect, no effect on the methyl group. The methylene group is slightly enriched. But if we look at the 2 different positions in the methylene group, the Pro-R and the Pro-S, we see that one of these is very strongly enriched, whereas the other is not. In other words, biochemistry has transferred the hydrogen to a single prochiral position in the molecule. So this can tell us a great deal about how hydrogen is being fractionated within organic molecules. And this is an example to show you how an enzyme can be very responsible for enriching or depleting hydrogen site-specifically in this way. We can go on a little bit further and we can actually put some numbers onto this because we're working in a quantitative NMR. We can make quantitative measurements. We can measure the surface areas, as I showed you, under the peaks. And so we can actually calculate what we call the transfer coefficient. Because, of course, some hydrogen positions can have more than one origin of their hydrogens, as you'll see in a minute. Very simple little equation. It's a bit of an x equals a plus x, y equals ax plus b sort of situation. It's a nice linear relationship. And basically we produce this transfer coefficient, which is the contribution of any deuterated position in the substrate to I, to any deuterated position in the product J. B is just the factor that adds up all the other sources that you can have, water, other positions in the substrate, etc. And just to give you some idea of how this actually pans out when we put the numbers into this, don't worry too much about the numbers here. Here's your transfer coefficient. Oh, it's blue that's missing as well. That's in blue. That's a sort of blue. Okay, so for the methyl group, here's your 1 deuterium, goes straight through, no metabolic problems. We get a very high conservation. Now, the 2 position here, we have a transfer coefficient which gives us about 60% incorporation, and that's because the 2 position is moved intramolecularly, and there's a certain amount of exchange with water in the glucose 1 6-phosphate to fructose 1,6-phosphate isomerization. The interesting one that I really like to look at again is the 4. Here's the 4, and this goes through at about 20, so obviously again that is very diluted with water. Now, how do we explain all that? So to the 1, here we can follow this through glycolysis now. The 1 goes through here, and it goes straight into the group, the methylene group here. The 4 goes off during glycolysis into the NADH pool, and that comes back in from the NADH pool through ethanol dehydrogenase into the methylene group in both cases. So we see how enzymes are responsible for specific hydrogen positionings, either because they don't touch them at all, either because they move them from one position in the substrate to another substrate position in the product. So that AGI for the 1 position, which now becomes the methylene group, of course, has 2 hydrogens in the substrate coming into it, as well as a hydrogen from water. So our water hydrogen comes in here, the underlined water. So again, the effect of water, if the water we're working with is very depleted, then of course this position will be very depleted, and this whole methyl group will, when we average out the 3 hydrogens, and I'm afraid we can't separate the 3 hydrogens in the methyl group, will also be very depleted, likewise enriched. Okay? We now go back and look at Lutonostock, which I'm going to show you some data for in a minute. We see, just to remind you, that this goes through a rather different pathway. We're going to compare the ethanol produced by glycolysis with the ethanol produced by Lutonostock. The thing I want you to note here particularly is that in the methylene group here, both hydrogens have been produced from glucose, introduced from glucose. Neither of them come from water. And we can show that quantitatively, and I'll just run you through the 2 little experiments. Here's the the CH3 group of our ethanol. Here's water, this is the control, water that's been slightly enriched again, a little bit of extra water added in, and we find that group has been enriched with water as we would expect. We also find that it's been enriched by the 2, because this is Lutonostock, not yeast, there's no enrichment from the 1 position, because we know that the hydrogen there is lost to water, and the carbon to carbon dioxide. So neither the 1 nor the 3 position. Nor, of course, you'll notice the 6, because that goes into the lactic acid. Quite a different profile. We look now at the methylene group in the same way. Again, we get incorporation from water through metabolic exchange. This time the 1 and 3 are what are enriching the position, but as you see, they're both, the 1 and the 3 enrich it. And the 2 doesn't, and neither does the 6. The problem we then were faced with was that if we looked at the enrichment in these 2 positions in the methylene, we know that they're both enriched from the 1 and from the 3, but we see that the ratio of the enrichment is really not the same. In the 3, from the 3 position of glucose, the enrichment is pretty well comparable, but from the 1 position of glucose, one group is enriched very much more than the other, relative to the control value here. And so the site 1 was transferred apparently preferably to the pro-R position, and the site 3 was transferred pretty much the same to both positions. So here we have a situation where apparently we have 2 hydrogen positions, 2 hydrogens from the substrate, but in fact the degree of enrichment, or the degree of depletion in this case relative to the substrate, is not the same for the 2 hydrogen positions in the methylene group. So this is just running through to show you that we have 2 reductions. We have one here which uses NADH to introduce one of the hydrogens, and the next one that uses NADH to introduce the second of the hydrogens. And that these NADH molecules obtain their hydrogens from the 1 and the 3 positions of glucose through the 2 steps earlier on in the pathway, the glucuronate reduction, and the essential thing being that these 2 enzymes can both use NADH and NADPH as the acceptor of the removed hydride. So what we ended up doing was we now have to do some enzymology. And in fact, what we now come to is something that has nothing at all to do with isotope effects. This is not an isotope effect at all. It's not to do with deuterium depletion at all. What it's actually due to is cofactor usage of the different enzymes. And what we now find is that if we look at the cofactor usage of, in brown we have the enzymes that are taking the hydrides off, in blue we have the enzymes that are putting the hydrides back on again, we see that for the glucose 6-phosphate dehydrogenase, which removes the 1 hydrogen, it's not too worried about what its cofactor is. It uses both NAD and NADP as receptor, a little preference for NADP, but not huge. If we look at the second one, however, which removes the 3 hydrogen, or deuterium, we see that NAD is very much the preferred substrate relative to NADP. Glyceraldehyde 3-phosphate, which removes the 4 hydrogen. We see, again, NAD, making NADH, is very much the preferred cofactor. We now come down to looking at these enzymes that are using these cofactors to transfer the deuterium or the hydrogen back into the substrate, into the product that we're looking at. We see straight away that the lactate very much strongly uses NADH, but the ethanol dehydrogenase very much uses NADPH. So we have, between lactate and ethanol, we have a very strong difference. But of course the 2 enzymes we're interested in are the aldehyde dehydrogenase and the ethanol dehydrogenases. And we find here that the aldehyde dehydrogenase, which reduces that first hydrogen to one of the positions in the ethanol, has a strong preference for NADH, whereas the ethanol, as I say, has a strong preference for NADPH. So that deuterium distribution that we saw in the ethanol, in the 2 prochiral positions of ethanol, in fact appears to be very dictated by the cofactor usage of the enzymes involved in introducing that hydrogen. So that's one story about how hydrogen depletion can be regulated in metabolic pathways. So we have a combination, really, of the deuterium pattern in the substrates, the pathway exploited by the organism, and the properties of the enzymes in that pathway that are exploited by the organism. I haven't talked about kinetic isotope effects, particularly in this case. This is purely looking at observational information on deuterium levels and the deuterium depletion that we see in different positions. Now I want to move on to a slightly more complicated system now, for the 10 minutes or so that I've got left, where I'm going to look at deuterium distribution in fatty acids. And this is much more complicated, but it's really very similar because, of course, we all know fatty acids are essentially made up of these 2 carbon units, which derive from acetate, which is very closely related metabolically to ethanol. And we have just 2 little things we have to bear in mind when we're looking at fatty acids. We have what I'd call the even positions, which comes from the methyl group of acetate. And we have the odd positions, which comes from the COOH, the CO group of acetate. So the methyl group has potentially hydrogens from acetate itself and from water, whereas the odd site hydrogens have to have been introduced into that molecule from some other source than acetate, NADPH in this case, and water, as I'll show you in a minute. We also have the situation, of course, where if we introduce a deuterium into one of these hydrogen positions, we end up again with a prochiral carbon center, just like I showed you for ethanol, and therefore these hydrogens are not the same. Now, here's the spectrum again, the NMR spectrum. It's just like we looked at for ethanol, except with a few more carbon centers in it. Here we have pyridine, which is our reference molecule for fatty acids, partly because TMU falls unhelpfully about here in the spectrum. 2 things I'd like you to note. First of all, some positions, like the methyl group, for example, are fully resolved in this spectrum. Other positions, like the most interesting ones, of course, the desaturation positions, 9, 10, 12, and 13, are completely unresolved in the deuterium spectrum, which gives us a bit of a headache. However, further techniques, which are rather more sophisticated NMR, which I'm not going to go into at all in detail, do allow us to get much, much higher resolution on these. I'll just show you one spectrum of that in a minute. But what I want you to start off with is just to look at what we get from that NMR spectrum that I've showed you. And here's looking at linoleic acid, or methyl linoleate, 2 double bonds, a number of CH2 groups, isolated from 3 different plant species, doesn't matter what they are. We see 2 features here. We see, first of all, the feature, well, we see 3 features actually. They're all rather similar. That's quite interesting itself. Metabolically, they're all rather similar. We have an alternating pattern between the odd positions, which tend to be depleted relative to the even positions. And we have 2 positions which are particularly interesting, the 9 and the 13 in the double bond, the 2 double bonds, 1 in each double bond, which is very depleted relative to the rest of the molecule, and is also very depleted relative to the 10 and 12 positions, which are also found in the double bond. So this isn't simply the fact that it's been reduced and had a hydrogen removed, because otherwise, of course, you would see exactly the same effect in the 9, 10, 12, and 13 positions. So here we have a metabolic enigma, which I have to say we completely misinterpreted to start with, and indeed published our misinterpretation. It's always nice to publish a misinterpretation. It gives you the opportunity to publish a second paper correcting your first paper, which we were also able to do. Okay, what's the cause of this? Well, we've lost the iron here, I'm afraid, completely, but imagine, okay, oh, you can just see it here. If you imagine there and there are 2 huge iron atoms. What we have here is a 3-dimensional image of the only fatty acid desaturase which has actually had an X-ray crystal done on it, the delta 9, which is going to take 2 hydrogens off this bond. And as you can see, there are 2 hydrogens here and 2 hydrogens here. Now the iron center here, which is going to take those hydrogens off onto the oxygen atom that's attached to the iron, is orientated such that the hydrogens are taken off from the same side of the molecule on every occasion, except for the very low statistical probability that they're not. So essentially, the same 2 hydrogens are taken off. Now, I'm just going back. This is our clever NMR, where we can actually, by using rather sophisticated methods, separate out those 2 hydrogen positions in the methylene groups, in the CH2 groups. Okay, so here I'm just going to show you one part of the spectrum where it's particularly clear. And there are 2 things I'd like you to notice. First of all, here's the even position, a 16, and here's an odd position, a 17. We can separate those 2 prochiral hydrogen positions on the CH2 group, the long chain. And what we see is that in the even position, the 2 hydrogens have a rather similar deuterium content. The S is slightly depleted relative to the R. If we now look at the odd position, 17, the mean value is very much the same, but in this case, the R position is slightly enriched relative to the even position, but the S is very depleted. The S position is really very, very depleted. So here we have a situation where in the synthesis, the biosynthesis of the long-chain fatty acid, the reaction, which is an NADPH reductase, which has introduced this hydrogen, is very sensitive, obviously has a high kinetic isotope effect selecting against deuterium, which means that this hydrogen position is depleted in deuterium. Okay, so the 2 messages are particularly that the S enantiomer is impoverished compared with the R, and that the odd site is very much more so than for the even site. Now let's go back to the biochemistry, and we know that this enzyme, not our work, previous work, John Shanklin and other people who are real experts in this area, have shown that when the desaturase attacks its substrate, in this case we've got both the desaturases, we're going through here to our linoleic acid, it is always the pro-R hydrogen that is removed, and this was actually shown originally by Cornforth and co-workers in the Nobel Prize for the steroid condensations. We're removing the pro-R very specifically on each case. What we're going to do is we're going to end up with the 9 and the 13 positions retaining the pro-S, which was very impoverished. And we're going to end up, of course, with the 12 and 10 positions retaining the Pro-S, but in the even positions, that was not particularly impoverished. So here we have the explanation for our data. The 9 and the 13 is the residual hydrogen that was already very impoverished in the substrate. The 10 and the 12 are very much average values, normal values for the compound, and therefore they have not shown this same impoverishment. So this is not a kinetic isotope effect, which was our initial interpretation of the data. This is in fact due to the hydrogen level in the substrate. A little bit like I showed you for glucose, that when the hydrogen varies in the substrate, that variation can be reflected in the product. But in this case, the enzyme has removed the rich hydrogen in this position and left the depleted hydrogen, removed the rich hydrogen in this position and left the depleted hydrogen, whereas in these positions it's removed the hydrogen which is almost the same value as the one that remains behind. So here we have the explanation for this, which this took us nearly 10 years to understand this, to be able to explain why we see this depletion in only one position on the double bond. So what we've managed to explain in this work is exactly that, why we have different distributions in the Pro-R and the Pro-S positions, and in fact we do probably have a secondary kinetic isotope effect as well during desaturation, but that's another piece of work. What isn't explained, really, is why is the deuterium depletion different at the even and the odd methylene sites in the first place? Is this due to different hydrogen origins? Is it due to kinetic isotope effects during elongation? And I'm running out of time. Yes?
Go ahead. Yeah.
Well, I can either answer those questions for you or not, depending on whether I have another 10 minutes.
Go ahead.
Okay.
Go ahead.
Yes? Vote show of hands? Not me. Okay, well, I will go on and answer those questions, but I thought I'd probably better stop there. So we have four types of sites that exist along the chain. I'll just come back to this picture again to remind you of the odd sites where we have hydrogens that have come in through the reduced cofactors. And we have hydrogens which have come either from water or acetate. Now bear in mind already, of course, that I've shown you that the reduced cofactor pool exchanges its hydrogen to some extent with the medium water. Okay, well that was going to be the end, so I'll acknowledge Frederick Soddy at this point. Thank you, Frederick Soddy, for giving us all a job by having discovered isotopes, and of course he was awarded the Nobel Prize for Chemistry in 1921. I'm ashamed to say the house he was born in Eastbourne in England is now a nightclub. It shows how Britain acknowledges its scientists. Okay, let's go back to fatty acids. Now, the experiments that we did, again, involved a little bit of enrichment to get our depletion, if you like. We took, in fact, a fungus, which grows in liquid culture, Fusarium, and this produces a great deal of fatty acid. It's an oleaginous fungus. It can produce up to 40% of its dry weight as fatty acids, and therefore it's a nice organism if you want to do controlled experiments with fatty acids. It's also horrible to handle. We can take natural abundance, just like I showed you before with the yeast and the leuconostocs. We can take water and add a little bit of deuterated water to it. We can take glucose and add a little bit of extra deuterated, site-specifically deuterated glucose to it. And then we can look at the distribution of deuterium within the fatty acids. And what we see is, again, that we get a distribution which, if you look at perhaps the green line here to start with, it's very similar to what we had before. Here's with the enrichment in the 6,6 prime position. Some positions are enriched more than others. So NAD, sorry now, is not NAD, nicotine adenine dinucleotide, it's natural abundance deuterium. I should have changed that from my colleague who uses that incorrectly. So these chemists, they don't understand biochemistry. Anyway, here's the natural abundance values, and as you can see, there's really some positions have been a little bit enriched, as we might expect, and others have not. Water has a big effect, and here we see a huge impact of water on this up and this even-odd effect. So water is having a much bigger effect on the even positions than it does on the odd positions. Glucose 1 also has enriched quite considerably the odd positions relative to the even positions, but it's not terribly clear when you just look at the ordinary NMR spectrum. But if we use the NMR spectrum, that allows us to separate in much higher resolution these positions, then we get a lot more data out of it. But we do see here that the deuterium is incorporated site-specifically and is related to the source of hydrogen that's been available. So we can do exactly the same thing. We can go through this, we can measure the areas under all the peaks, we can then calculate these transfer coefficients, and we can come up with a sort of map, if you like, for deuterium origins into different positions. So if we take the terminal methyl group of our fatty acid, we can actually get these transfer coefficients from water. We see there's really quite a lot of water goes into that. About 60% is coming from water. Ignore the acetate for now because that was a bit over-enriched. We can see, as we would expect, the methyl group is getting its hydrogens rather efficiently from glucose, which is slightly enriched in deuterium in the 6 positions. So it's just like the ethanol, of course, which went rather directly into the 6th position. We can also see that there's a small transfer from the 1 position. And that was not so easy to explain to start with. If we look now at the pro-R position, we can see again a huge incorporation from water. Now this is the position which should really be getting its hydrogens from water and through glycolysis. We have a small link with the 6,6, and we have a small link with the 1. So we can see again where this hydrogen is actually coming from. And again, the link with the 1 is a little bit unusual. We can do the same thing again for the other hydrogens, for the Pro-S. And now we see with the Pro-S, absolutely no link whatsoever to the 6 or the 1 positions of glucose. And a very, very large amount of this has actually been introduced from water. And finally, looking at the Pro-R of the odd sites, we have much less coming in from water and really a lot coming in from glucose 1. So the Pro-R of the odd sites and the Pro-R of the even sites are not really showing the same links through to glucose and water. If we look at the Pro-S of the even sites, water is contributing very little to this hydrogen position. And glucose 1 is also contributing, and in fact so is glucose 3 and 5 that we were not able to do the experiments with. So what we can see, basically, the take-home message here, if you like, is that we can show the origins, or at least some of the links through to the hydrogens in glucose in the different positions of the fatty acid, depending on, it depends on very much on the biochemistry and the availability of deuterium in those different positions within the substrate glucose and within the water of the fermentation medium. The other thing that clearly is that the sites in the odd positions and the even positions have different links through to those the potential origins, and that can be traced back, and I won't go into the details of that, to the positions in the glucose as we would expect. Through glycolysis, this goes through to acetate, where the 1 and the 2 are going to be incorporated into that methyl group, as we would expect them to, and the 3rd hydrogen to be coming from water. The 2,6 positions will also come into the acetate-methyl group, and that's exactly what we saw, those going into the methyl, the even positions in our fatty acid chain. But what is also clear is that, in fact, we have 2 pathways going on at the same time in this organism, which we didn't expect, was that we'd not only had glycolysis producing acetate by the classic glycolytic route, but we also had the reductive pentose phosphate pathway running, which, if you remember, it had quite a different deuterium isotope pattern in ethanol. And so the reductive pentose phosphate pathway is clearly transferring glucose from the 1 and the 3 positions, deuterium from the 1 and the 3 positions of glucose. They're transferring that into the renewed reduced nucleotide pool, so that the reduced nucleotide pool is getting enriched from the 1 and 3 positions, and hence, of course, that's coming into the fatty acid chain through the reductases in the fatty acid synthase cycle, whereas the 2 position is coming into the methyl group classically through the production of acetate from the reductive pentose phosphate pathway. So again, we can see here that the levels of deuterium incorporation or the levels of deuterium depletion that we're getting within different positions of the molecule can lead us to interpreting the metabolic background. And indeed, in this particular instance, just showed it quite clearly that we had 2 metabolic pathways providing our source reduced nucleotide, which we were not expecting to find at all. Okay. Oh, well, there we are. There's a little animation. So this hydrogen here, in particular, on the glucose 6-phosphate goes into the nucleotide pool, which then comes into the even positions. So we can basically say that these odd sites are getting their hydrogens from the 3 through the pentose phosphate pathway. They're getting it from the 4 through the glycolytic and the pentose phosphate pathways. Whereas even sites getting almost certainly would be incorporating the 2. That was an experiment which we did 3 times, and every time the culture got contaminated, so we gave up in the end. And that the Pro-R is also getting it from the 2 position, but with a great deal of exchange with water, as you saw earlier. Nevertheless, theoretically, we should not expect to see that difference between the Pro-S and the Pro-R positions, which in fact we did observe in these fatty acids. So therefore, it does appear that there are some kinetic isotope effects taking place. We have not been able to do the experiments to test those out, but there clearly are enzymic factors in addition to glucose deuterium availability in the fatty acid synthesis system. So you can look at other fatty acids as well, and I won't go into any detail on that. That we've looked at the hydroxylated fatty acid ricinoleic acid. We've looked at the epoxidated vernioleic acid, and we can do exactly the same sort of approach. And we can actually show again that there are deuterium depletion effects in these positions, which can be related to the metabolism that's taking place. So we should now see Frederick Soddy, but we've already seen him. What I really wanted to convey to you this afternoon was the extent to which deuterium availability or lack of availability is clearly going to have major impact on different positions within different molecules. So you can't just say, as we heard earlier, if water is labeled deuterium, water is labeled deuterium or not. That's true. If we take glucose, if we take ethanol, if we take any of the metabolites that we're interested in, which may have a function within metabolic pathways or be end products of metabolic pathways, deuterium is not naturally distributed within them. So a deuterium in one position may have a great impact on the action of that molecule or the metabolism of that molecule, whereas a deuterium in another position in that molecule may have no effect at all. So if you use mass spectrometry, isotope ratio mass spectrometry, to measure the mean value in deuterium, or indeed in carbon-13 for your molecule, you get a mean value. But the critical thing is that if you've got, like a fatty acid, 30-odd hydrogens within it, and one of those is very depleted or very enriched, you'll lose that information completely in a mass spectrometric analysis. We work with people in pollutions. They say more than 10 carbons, you really can't distinguish between one that's depleted or one that isn't with a hydrogen position on it. So what we're moving into here is having to say that site-specific deuterium distributions, depletions within molecules can also have major impacts potentially in metabolic processes. Thank you very much for your attention, and thank you again for the invitation to present this work to you.
Please, any questions?
Please make me clear. Two slides before. This is the main question. How water products can introduce in the proteins of belongs to carbon?
This is experimental data. Yeah, this is obviously a very large summary of the experimental data. It was made by taking our bacterium, our culture of fungus, growing it in the presence of glucose that was slightly enriched, and then isolating the fatty acids, separating the fatty acids by chromatography, bulk chromatography, well, the methyl esters actually were separated, isolating the methyl linoleate, then measuring by NMR the deuterium levels in each position within the methyl linoleic acid. Okay? And then you can say, well, you could, relative to the control, of course, we go back now. So all the values.
This is the result of biochemical process.
This is purely a result of biochemical process. Yes, yes. So all the value we have here now is this level of incorporation of an individual hydrogen from an individual position in glucose, or from water.
This is very interesting. So do you know if there is any biological role of carbon-9 being so different from the rest of the saturated or unsaturated fatty acids? And the reason why I'm asking this is that actually peroxysomal beta oxidation stops at octanoic acid, which means that once the fatty acid chain loses its 9-10 acetate units, it has to be transported from the peroxisomes to the mitochondria for complete oxidation.
Yeah. Yeah.
And my question is if there's any biological signal or any specific role of this particular carbon being so different as far as its chemical properties. That's one of my questions. The other question is...
I think you're probably the person to answer that.
I'm sorry?
You are probably the person to answer that question. No, I don't think so. I think that's probably more a biochemical... I mean, I would suspect that's more a biochemical factor. That is to say that the enzyme, the beta oxidation process cannot handle the small fatty acids, can it? So
we don't really know why the peroxysomal beta oxidation process stops at the octanoic acid or the 8 carbon unit. We don't know. And I think this is just really interesting to kind of correlate the two findings, yet we don't really know why peroxisomes stop at 8 carbons.
My guess would be that it was a substrate related problem, not an isotope related problem. That's to say that the beta oxidase system can't cope with such a small substrate.
Well, if that's true, well, let's say say it has a biological role. Maybe the acetoenzyme A, which is actually producing an octanoic acid and an acetoenzyme, which maybe it has a different dynamic simply just because there's a different hydrogen mass distribution on the acetate unit, and maybe it's just some kind of a signaling pattern, or there's actually a true kinetic, biological, or enzyme kinetic event related to that oxidation of that carbon. But this is just obviously just kind of a hypothetical kind of thought process here.
Well, you could test that by making chemically synthesized fatty acid, either enriched or depleted in deuterium in the 9 position. We can make 9-10 fairly easily, and then using that as a substrate, I suspect it would probably not have a major effect. I would think it's more likely to be a substrate. I mean, one has to wait for the X-ray structure and then do the modeling, I guess.
I just find this particular very interesting, and obviously
it is.
Being a biologist and biochemist, I think it's just intriguing.
I think that would for me that would, my first instinct would be that would fall into the type of interpretation I was giving you about the Lucas-Nostock transfers, why the differences in the 2 positions, that it was actually that was a biochemical problem. where the biochemistry was such that the hydrogen usage caused differences. I would think that would be a biochemical
we know
problem.
that octanoic acid is actually a very fit substrate for mitochondrial beta oxidation. So from then on, the energy production process is extremely efficient.
Yeah.
And this is how we actually measure the octane number of the gasoline.
Right.
So when it comes to like ancient biological oxidation, obviously where there is no sunshine, just like for the proximal beta oxidation process, that seems that that's the chain length that has to be transferred to the mitochondria for a more efficient energy production process in mammalian system or biological systems. Obviously, this is just interesting.
But then you ask the more fundamental question: why do you actually have to have the transfer from the peroxisome to the mitochondrion? I mean, why doesn't it all happen in the mitochondrion? And
I think eventually the question is why there's a cutoff point where actually you find the hydrogen, which is actually most critical, most different
that's interesting,
from
isn't
the
it?
rest of the molecule. And again, this is just I don't have any other proof of this other than just practically just generating hypotheses here. The other question is, for the mammalian system, linoleic acid is obviously an essential fatty acid, and oleic acid is a non-essential fatty acid. So when it comes to mammalian systems, let's see, related to cancer studies, obviously the linoleic acid has to be obtained from the oils that are produced in plants. Yet in mammalian systems, oleic acid can be produced from acetyl coenzyme A, as you were pointing out how those go. Do you have any studies that are taking cultured mammalian cells and see how the oleic acid look in those systems?
No, that would be very nice to do. We've not done mammalian. We have looked at avian, and you get very much the same distribution pattern in the fatty acids.
I'm going to talk about tomorrow about the fatty acid synthase structure in mammalian systems versus fungus. It seems that you're working with the rigid type or the type 2 fatty acid synthase in fungus. The mammalian system is very different from this, very sensitive to, and I'm going to talk about this tomorrow. So maybe there's some other interesting thoughts that come out from there. Thank you.
Thank you.